So continue being on time. We have our second speaker is Dr. Alicia Widge. It's my pleasure to introduce her. She is the Associate Chief for Clinical Research and Development for the Clinical Trials Program at the Vaccine Research Center at the National Institute of Allergy and Infectious Diseases at the NIH. In addition to providing overall scientific and trial development guidance, she actually served as a principal investigator for numerous phase 1 clinical trials studying novel vaccines, including the phase 1 clinical trial of the COVID-19 Moderna vaccine. She will be talking to us about SARS-CoV-2 vaccine trial research. Welcome.
Thank you very much. It's really looking forward to speaking with all of you today. As a former children's national resident and now someone working at NIAID, this is a special pleasure. So really looking forward to it. So I'm going to talk today, if I can get my slides to advance, about COVID-19 vaccine clinical research. So I'm going to be focusing on mRNA vaccines because that's the only vaccine that's currently authorized for children in the area of my primary research. And I'm going to talk a lot about our phase 1 trial results that enabled the later efficacy trials and eventual emergency use authorization of these vaccines. I think it's great to understand this initial phase 1 data because that's what leads us into dose selection and really helps understand pediatric trials that are ongoing now and vaccinations that are being given to children. I'll then discuss the existing data on mRNA vaccines in a pediatric population and the expected timelines for when they might be more widely available. So to start, I want to take us back on a little bit of a history lesson, if you'll oblige me. So going back to December 31st of 2019, so that's when the first cases of a respiratory virus outbreak were occurring in Wuhan, China. And I remember being at meetings at the NIH on January 6th and sitting in a conference room with Barty Graham and others where we said there's an outbreak, it seems to be a coronavirus, seems to be isolated for now, we should think about making a vaccine. The VRC and NIAID had an existing collaboration with Moderna. We had been thinking about coronaviruses and mRNA vaccines actually for many years and really knew that mRNA vaccines had a key potential to be used in pandemics. We'd actually had a clinical trial plans that were sort of ongoing for an mRNA vaccine to other viruses. And so we were able to leverage its existing collaboration and the VRC's experience in working with coronaviruses to start work on this immediately. So the viral sequences were published on January 10th. And in just 2 days, the VRC and Moderna decided on the vaccine sequence. So we were able again to do that in record time because we had so much experience in this area. That kicked off a whole chain of events that all occurred concurrently. So to start with, Moderna started GMP production of the vaccine, and then simultaneously, DMID, the Division of Microbiology and Infectious Diseases at NIAID, started the clinical trial and regulatory processes. So they were the sponsors of the phase 1 clinical trial. At the same time, all of the preclinical work is being done. We're submitting for regulatory approvals. It's all happening very quickly. And the clinical trial starts just 66 days later on March 16th. So this is so remarkable. And I know we touched on this in the first session, but for those of you who have done a lot of clinical trials and research, this is just really mind-blowing. A process that would typically take years was able to be done in a couple of months. So from the time we decide on the sequences to the time the first trial participant is injected is just 66 days. March 16th is an interesting date. So some of you may remember, at least for the federal government, that was the first date that we shut down and said, only essential personnel only. I think that's when COVID really ramped up in the US and became really real and really scary to a lot of people. And that was the same day that we injected the first clinical trial participant. It's hard to believe it's been over a year. So that enabled then the phase 3 to start just a couple months later, and then emergency use authorization to be issued on December 18th. So this is just a reminder of our critical spike protein. And this atomic level structure was really key to developing the mRNA vaccine and an understanding of it. I don't know if anyone knows this, but the reason it's called mRNA 1273 is because there's 1,273 amino acids in this spike protein. So that's my fun bit of trivia. And here in green is the key receptor binding domain. And again, scientists understanding the structure at the VRC was key to us then developing an mRNA vaccine. So the general principle of this being, we take mRNA encoding for this spike protein, then inject it and take it up into muscle cells. which make the spike protein from the mRNA, and then we generate an immune response. So this is really great for a number of reasons. And the first one is that this enables us to display the antigen in its sort of natural conformation. So you have its intact conformation, you're displaying the receptor binding domain the same way you would see it from natural infection. And that enables you to generate a really robust and a really specific immune response. The other great thing about this delivery system is you get a capacity for really rapid, inexpensive, and scalable manufacturing. So mRNA vaccines are relatively cheap to make, and they're really fast to make. You can also leverage and change the mRNA. And so you get sort of this plug and play system that we knew would make this really great for a pandemic. So you're getting antigen presentation in vivo that mimics natural infection, and you're able to do it really quickly. And I'll talk about this a little bit at the end too, but because mRNA is able to be that kind of plug and play system, we now have mRNA vaccines for some of the common COVID variants that we're testing in clinical trials right now. So you're able to sort of change it with time, which makes it really appealing for a pandemic response. So this was the first ever person to receive an mRNA COVID vaccine at a Kaiser site in Seattle. And so I'm going to talk about our phase 1 study design. We started initially with 45 subjects aged 18 to 55. That's typical for our phase 1 studies in healthy adults. As we were enrolling them, more data was coming out that older adults had much more severe COVID infection. And so we realized we needed to broaden our population to study this vaccine in older adults as well. So that began shortly after. There were 3 sites for our initial phase 1, Seattle, Atlanta, and then our site at the NIH in Bethesda. So this is our phase 1 study schema, and also an excuse for me to show you the photo of us injecting our first participant at the Vaccine Research Center. And there's me overjoyed and our nurse Whale injecting our first vaccine recipient. So our initial cohorts were 18 to 55, and we tested in a typical dose escalation design, doses of 25, 100, and 250. We tested similar doses in older adults, and then we also explored a dose of 50 micrograms. So what we saw is that 100 micrograms really seemed to be the sweet spot. Doses of 25 and 50 are immunogenic, but doses of 100 are even more so and are very tolerable. Doses of 250 take all the side effects of 100, but they're a little bit more amplified. And so a dose of 250 did not seem quite as tolerable for the general population as 100. And so this is how a dose of 100 then was selected for studies down the road. So I want to talk you through now some of our initial binding and neutralizing data from the phase 1 trial. So here on the left, this is an ELISA looking at binding antibodies to the receptor binding domain. And on the right, these are neutralizing antibodies measured by our pseudovirus neutralization assay. So we have titers on the y-axis and days post-vaccination on the x. And I'm showing you here doses of 25 and 100 micrograms. Highlighted in the red boxes are the doses in the increasing age cohorts 2 weeks after the boost dose. And what you can see, looking here over here on the right, this is the 18 to 55, so the youngest age cohort in the phase 1. These are the over 71-year-olds, so the oldest, and then the 56 to 70-year-olds. And they all generated really robust binding antibody responses across all the age cohorts. And it was a similar story with the neutralizing antibodies. They looked really similar across the age cohorts, and they looked really robust. This was great news because, as you know, older adults don't always generate as strong an immune response. You can sometimes see some immunosenescence, and we did not see that here. And even more excitingly, when we compared it to a range of convalescent controls, the responses from the vaccine were in the upper quartile of those generated in convalescent controls. These convalescent controls were also collected at our clinic at the Vaccine Research Center, and it was no easy feat to get these. So we were collecting these back last February, early March, when it was hard to even find a COVID test to confirm that someone had COVID to call them then a convalescent control. So this was a difficult panel to get, but has turned out to be really useful as we've gone on to analyze the data. We also saw cellular responses against this vaccine. So here looking at responses from Th1 and Th2 cells. And so we saw a strongly biased expression of Th1 cytokines, as you can see here in both the 100, mostly in the 100 microgram compared to the 25. And we didn't see any Th2 biased cytokine responses. This was really critical. This was at a time in vaccine development where there were still some questions and concerns about whether this vaccine could lead to vaccine-enhanced respiratory disease in which you might see a more Th2 bias response. And so we did not see that. And so this was great news that the vaccine was generating cellular responses as well. We now have data published from the phase 1 trial out to day 209. So this is 6 months after the boost dose. Here I'm showing you our live virus neutralization assay. And we found that all of our neutral, our pseudovirus and our live virus neutralization assays and our binding antibody assays all correlate exceptionally well. This assay is our most sensitive, but what we saw here, so we have ID50 neutralization titers on the y-axis and then days post-vaccination on the x, again, going all the way out to day 209, so 6 months after the boost dose. We have 3 age groups, the 18 to 55 in blue, 56 to 70 in orange, and then in gray, the greater than or 71-year-olds. And so this was tested in a random sample of 33 participants from the phase 1. And we saw that all of them have detectable neutralization activity out to day 209, which is great news. You do see an initial neutralization response after the first dose, but there is a clear pronounced response after the boost. And you see a modest decline, which again is expected, but overall titers do remain elevated in all the age groups. Highest in the youngest, which is not unexpected, but this is great news that all participants have detectable titers out through day 209. So even more excitingly, we've, based on our phase 1 data, studied neutralization responses against variants of concern. So this is on our pseudo-neutralization assay, and this is looking at 8 volunteers per age group, all of whom received 100 microgram doses of the mRNA 1273 vaccine. And what we see here is that neutralization of variant viruses is sustained even through 6 months after the boost dose. So talking through here, this is our 18 to 55-year-olds, the youngest cohort, 56 to 70, and then greater than or equal to 71. And on the left is the D614G, so what was the most commonly circulating variant, B117, which was the variant that originated in the UK, and then B1351, which is the variant that originated in South Africa. And so we see cross-reactive neutralizing antibodies for all of these different variants. B117 has the least impact on neutralization, and B1351 has the greatest impact on neutralization, but you do still see some activity. So we see binding and functional antibodies against variants in almost all of the subjects, albeit at lower levels for B1351, going out to 6 months, but they are there. This other thing I want to point out here is we don't really see a lot of cross-reactive neutralizing antibodies after a single dose. So it does seem like, at least based on this data, that the boost dose is critical. So if you look here, day 29, so this would be after the prime, and then 43 after the boost. And the boost does appear to be critical for generating these neutralizing antibodies, especially against these variants of concern. So overall, to summarize our initial phase 1 data, mRNA produced high levels of binding and neutralizing antibodies at a dose of 100 micrograms 6 months after the boost, including to common variants of concern. We have ongoing studies to monitor the immune response beyond 6 months, as well as determine the effect of a booster dose to extend duration and breadth of activity against emerging viral variants. So DMID is now sponsoring another study, so sort of an add-on to our phase 1, where we're studying a booster dose of the original mRNA 1273 in this initial population to test both safety and immunogenicity of the original booster dose, and then a booster dose to variants as well. So we have a mRNA variant against B1351 that's being tested in a phase 1 clinical trial right now. We have data to share on that soon. So I think we're all familiar with the Kaplan-Meier efficacy curves from the phase 3. So our phase 1 work was then rapidly escalated into a larger phase 3 trial conducted by Moderna and Pfizer's as well. So similar mRNA vaccine design. And of course, we know there was clear efficacy of the vaccine, well over 90% for both of these. It is interesting that we do see efficacy after even a single dose of both of these vaccines. And efficacy is somewhere around 80% when you look at the data thus far. it's interesting when you compare that to the fact that we don't see really high neutralizing antibody responses after a single dose. So it does seem like the second dose is important, but we do see efficacy with a single dose as well when we look at the phase 3 data. So I'm going to talk now, it's the remainder of time talking about pediatric trials with both of these vaccines. So starting with the Pfizer vaccine BNT162b2, since this is the only vaccine that has emergency use authorization for those over 12. So this data came from 2,260 adolescents aged 12 to 15, and they had 100% efficacy in this trial measured as confirmed COVID disease greater than 7 days after the second dose. Their design was 1-to-1 30 micrograms of the vaccine versus placebo. They had a really favorable safety and side effect profile. It was very similar to what they saw in the older 16 to 25 cohort. Slightly more common was fever in this age group and the use of antipyretics. Everything else was really similar. So the most common being injection site pain, which was 79% after the first dose and 86% after the second. Fatigue, 60% after the first dose and 66% after the second, and headache, 55% after the first dose and 65% after the second. They didn't see any vaccine-related serious adverse events, including severe allergic reactions. They did not see any pericarditis or myocarditis in this population, but I will say this is a small number of participants. So when you compare the rates of allergic reactions that are being seen in the general population, you wouldn't necessarily expect to see it in the smaller population. So their next steps are they have an ongoing trial of 6 months to 12-year-olds. And this is done in a typical way we do pediatric trials, which is a age and dose de-escalation design. So they'll be testing doses of 10, 20, and 30 micrograms in the 6 months to 12-year-olds. And the idea being children often respond more robustly to vaccines than adults. And so you may not need as high of a dose in a pediatric population. We saw non-inferiority of the immune response in the 12 to 15 compared to the 16 to 25-year-old participants. And so here you can see the 12 to 15-year-olds with higher geometric mean titers than the 16 to 25-year-olds. So the Moderna vaccine is currently authorized for those 18 plus, but they just released results of their teen code study that also saw 100% efficacy measured as confirmed COVID disease greater than 14 days after the second dose and no safety concerns in their adolescent population that was 12 to 17 years old. There were just under 4,000 adolescents enrolled in that trial, and their design was randomized 2-to-1, 100 micrograms of mRNA 1273 or placebo. They started enrolling their kid code trial, which is their 6 month to 12-year-olds. That started March 16th and is ongoing in the US and Canada, and sort of similar to the design of the Pfizer pediatric trial. They'll have 2 to less than 12-year-olds who receive one of 2 dose levels, 50 or 100. 6 months to less than 2 years old will get one of 3 dose levels, 25, 50, or 100. And then they will use this initial data to select the dose in part 2, which will be the placebo-controlled expansion portion of the study. So to summarize, the safety and immunogenicity for adolescents with mRNA vaccines seems similar or more favorable than that in adults. We have pediatric dose de-escalation trials that are ongoing. The current expectation is that vaccines will likely be available pending emergency use authorization for 5 to 7-year-olds in late 2021 and for 6 month to 5-year-olds in early 2022. And we have trials in adults with booster doses and variants that are ongoing that, if successful, I think we can all anticipate would be taken into a pediatric population as well. So it's been quite a journey so far over the past year, and I think a lot more to come. So I just want to acknowledge everyone in our clinical trials program that's worked really tirelessly over the past year to get this vaccine trial enrolled and going. All my colleagues at the VRC and NIAID and DMID, the trial sponsor, and our very brave and wonderful study volunteers who volunteered to get an experimental injection of a vaccine in the early stages of a global pandemic. So we're very appreciative to them, and I'm very appreciative to all of you for listening to my talk. Thank you very much.